So for your question this week, you were given a data set from a study that looked at ischemic reperfusion injury and in particular liver damage. These were in wild type and TLR4 knockout mice. So you're given the text here that says that wild type or TLR4 knockout C57 black 6 mice underwent surgery to induce ischemia by occluding blood supply for 60 minutes, followed by 6 hours of reperfusion. There was also sham animals who had the laparotomy incision only. You're also told that a dose of 5 mg per kg of eritaran, which is the drug, or phosphate buffer saline, which is the vehicle, was administered intraperitoneally for about 30 minutes before the ischemic injury. And in addition to that, there was a separate group that had recombinant HMGB1 injected IP as well into sham animals. After 6 hours of reperfusion, all the mice were killed and liver tissue and serum were collected to assess liver function, cell death and cytokine analysis. So you were given 5 panels to look at, A to E, and you were asked to briefly describe the data in each of these panels. And then finally you were asked to draw conclusions about the mechanism through which the eritaran acted and to decide whether it was effective or not. So the diagram here shows the inflammatory pathway and in particular focusing on toll-like receptors. Highlighted here we have toll-like receptor 4, which is an extremely important member of the family, and it's involved in recognizing gram-negative bacteria. So it recognizes something called lipopolysaccharide or LPS, which forms a component of the gram-negative bacteria cell wall. LPS is normally recognized by an accessory protein, CD14, that brings it closer then to TLR4. TLR4 being a surface receptor, it communicates then with intracellular proteins, and it involves a phosphorylation cascade involving kinase enzymes that ultimately leads to the activation of the transcription factor, nuclear factor kappa B, and this involves the transcription and synthesis of new cytokines such as interleukin 6 and TNF alpha. And these are involved in inflammation. So the reason this is shown here is because normally this is involved in, this pathway is activated when we have an infection, but it can also be activated when we have injury or tissue damage. And in particular things like ischemic reperfusion injury often leads to a lot of inflammation, and this can exacerbate circumstances. It can often lead to organ failure and death. So the first figure we have here, figure A, shows the enzyme activity with the levels of serum alanine aminotransferase or ALT. Now this enzyme is normally used as a marker of liver damage because this enzyme is expressed highly in the liver. It's quite active in the liver, and it wouldn't normally be active in the serum. So when there's liver damage, the liver cells will burst open and they'll leak the enzymes into the blood. So if you get enzyme activity in the serum or blood, it's normally an indication that there's been some sort of liver damage. So this shows the serum ALT activity, and we can see the activity is expressed in international units per litre on the y-axis, and the groups are shown on the x-axis. So what I would suggest is that you'd maybe describe it as follows. According to figure A, ischemia reperfusion injury is significantly increasing the serum ALT levels or activity from less than 500 international units per litre to greater than 2000 international units per litre. And this might be indicative of liver damage. When eritaran is given, so group 3 here, it significantly reduces this level to below 1500 international units per litre, as you can see here. As a comparison then, the recombinant HMGB group, they had increased ALT activity to nearly about 2,500 international units per mil in the sham animals without the IR injury. Second figure, figure B, then shows the percentage of necrotic cell death in the liver following IR injury in wild type mice with or without the drug eritaran. So again, we can see the 4 groups and the percentage of cell death on the y-axis. So according to figure B, IR injury is significantly increasing the cell death in the liver from a low of about 5% to close to 20% then in the IR injury group. Again, this is quite indicative of liver damage. When Eritaran is given then to the IR mice, it significantly reduces the level to below 10%. And then as a comparison, recombinant HMGB1 by itself can increase the liver cell death to roughly between about 15 and 20%, so around 17% in the sham animals without IR injury. The third figure then, figure C, the serum levels of the pro-inflammatory cytokine TNF following IR injury in wild-type mice with or without the Eritaran. So we can see the TNF is on the y-axis and the groups are on the x-axis. So according to figure C then, the IR injury is significantly increasing the serum TNF levels from less than 100 picograms per mil to just around 400 picograms per mil. And this is a good indication of inflammation. When the Eritaran is given to the IR mice, it significantly reduces this level to below 100 picograms per mil. And then finally, as a comparison, the recombinant HMGB1 can increase the TNF levels to greater than 400 picograms per mil in the sham animals without the IR injury. Figure D then shows the serum IL-6 levels, and this is where we have the four groups again and the IL-6 measured in picograms per mil. So according to figure D, ischemia reperfusion injury is significantly increasing the serum IL-6 levels from less than 100 picograms per mil, you can see here on the first group, to greater than 500 picograms per mil, and this indicates inflammation. When Eritaran is given to IR mice, it significantly reduces this level to below to in around 100 picograms per mil. As a comparison then, the recombinant HMGB1 group, they see an increase in IL-6 levels to nearly 400 picograms per mil in the sham animals without the IR injury. Finally then, figure E, which is our fifth and final figure, shows the serum levels of HMGB1 following IR injury in wild type and TLR4 knockout mice with or without the drug Eritaran. So according to figure E then, we can see that the serum levels of HMGB1 in the sham mice, so this is the first two groups here, are around 10 nanograms per mil. So with the vehicle, it's just over 10 nanograms per mil, and with the Eritaran then, it doesn't alter it at all, so it stays pretty similar. The next group then, the IR injury group then, so IR injury significantly increases the serum levels of HMGB1 to at or above 25 nanograms per mil, and giving the Eritaran significantly reduces the serum levels to roughly just above sham levels. Okay, so there's a significant difference here. In the TLR4 knockout mice then, IR injury doesn't increase serum HMGB1 levels at all. So if you compare it here to the wild type animals that had IR injury, the levels are much lower in the TLR4 knockout mice. So giving Eritaran then as well to these animals doesn't affect the serum HMGB1 levels at all. So there's no further reduction in these animals. So having described the data, and that's all we had to do for the first 5 figures, we didn't have to infer any mechanism. So the final part is to infer the mechanism through which Eritaran might act and also decide whether it's effective or not. So what I suggest would be something like this. You'd say that IR injury releases HMGB1 into the plasma, so we can see that from the previous figure. So the IR injury seems to release the HMGB1, and we know from the previous figures as well that it can cause increased liver cell death, liver damage, and increases in pro-inflammatory cytokines IL-6 and TNF. We know from the other figures as well that the Eritaran is capable of blocking the increase in HMGB1 following IR injury and also reducing the levels of cell death and pro-inflammatory cytokines. So finally then, the conclusion would be perhaps the TLR4 may be the target for Eritaran, as the IR injury, as we saw in the last figure, doesn't increase HMGB1 levels in the TLR4 knockout mice. And also we noticed in this final figure that the Eritaran has no additional effects on these mice. So TLR4 could be important in terms of the mechanism of this particular drug. So finally then, when we ask is it effective, well, the answer would be yes, because it seems to significantly reduce the extent of liver damage, the amount of inflammation, and also the degree of cell death.